Martha Carlin is a dear friend of mine and a multi-time guest on the Sowing Prosperity podcast. She is all about those microbes, so lining out the gut microbiome and connecting it to these chronic diseases, metabolic issues that so much of the population faces. So in here, she gives a real solution for things to line it out and overcome the hump. Martha, so good to be visiting with you again. I love what you do and your relentless approach to understanding things. I greatly admire. So today I wanted to dive into your company, your work, and why that matters, especially for the epidemic of metabolic diseases, especially, type 2 diabetes is something that I'm seeing a whole lot. So if you will, can you give us a little bit of that introduction into you? Again, I think your third time on the Sowing Prosperity podcast, but that brief introduction to that, and then let's dive into Sugar Shift.
Sure. So just a brief introduction to me. I started the BioCollective back in 2015 after learning about the microbiome in 2014. And in 2014, I had been studying food and the complexity of Parkinson's disease and, trying to piece together all the factors because my husband has Parkinson's. So that was really driving me to teach myself science. And then when I learned about the microbiome in 2014, after reading Martin Blazer's book, Missing Microbes, I, about 6 months later, founded the BioCollective and started collecting people's poop and sequencing it to kind of look across diseases at the human microbiome to understand what do we share in common in health and disease instead of looking at single diseases, where could there maybe be shared lost metabolic function in the microbiome? So, we were very early pioneers in that field, and we are, I guess, one of the early pioneers that's still standing. There are not many of those early microbiome companies still around. And then over the course of that, through the data collection and analytics and building models, ultimately came to bringing on board Dr. Raul Cano, who's a microbial ecologist and he was one of the first people to sequence a lactobacillus genome. He was on the team that did that, and he's written several textbooks. I haven't run across anybody who knows microbes better than Raul. And so from that, we started to build our own collection of microbes and also work on formulas to put back functions that were lost, which led me actually initially to making Sugar Shift.
I love it. So the complexities of Parkinson's in a lot of ways mirror my experience with trying to understand cancer, because there's so many unknowns with the conditions. And so very early on, it became evident to me of the microbe connection to cancer, right? Because the microbiome does so many different things, and the complexities there are just wild. So why is there a way to kind of summarize the effect of why the microbiome has an effect on metabolism?
Well, so if you just look at, let's compare the number of genes with functions in our body so the human genome, the number of genes there compared to the number of metabolic and gene functions that come from the microbes in our body. So we're roughly, I think now they've come to a consensus, it's about 2-to-1 ratio of microbial cells to human cells. But in terms of gene functions, the microbiome is 200 to 300 times more genes than the human genome. So, as I have learned about it and thought about it over the years and looked at a number of what are called inborn errors of metabolism, I've started to think about those as, we start to recognize these problems with metabolism and we associate them with a human gene. But I think our microbiome is our primary functional performance in metabolism. It's breaking down our food, it's making our vitamins, hormones, neurotransmitters. There's a lot going on there. And when you lose part of that microbiome that has those functions, then you start to have these problems and somebody identifies this and says, oh, well, that's a monogenic disease. But really, you could put it back with a microbe that has that function in most cases.
So in the connection of, let's say sugar metabolism, for this, want to make it kind of more specific for, say, someone with type 2 diabetes. How can the microbiome affect blood sugar levels and helping to kind of mitigate the swings? Why is something like optimizing the gut so important?
Well, I would say, like, if you take a step back, you're setting up the terrain in your gut for certain bacteria. And that can be through, antibiotics or exposure to glyphosate. So you're getting organisms that are resistant to antibiotics, resistant to glyphosate. The American diet has a lot of fructose, sucrose, and those sugars, if you look at the profile of all the different sugars, they selectively feed certain microbes. So, I like to kind of take a step back and say a good part of it is the terrain that you're building by the things you put through the pipe. So then you're, losing those good functions or overpopulating with what we call are the bad guys. But, it's an interesting example is, some of the bad guys actually make some of the vitamins we need. So it's this not having a balanced and diverse community that's kind of keeping each other in check. Does that?
Yeah, I think that ecosystem analogy is vitally important. It goes back to maybe agriculture, right? When we look at how wild savannas and prairies and stuff it have been very diverse in the different forbs and grasses and trees and animals, birds, like all the different things going together. And through lifestyle, lack of sunlight, right? The overindulgence of these intense blue lights, glyphosate, all the chemicals. We have killed out a lot of the diversity. And so things just don't work right when we're not in line with nature. So what is going on specifically with sugar shift that is helping diabetics overcome some of the challenges? And then how have you been able to prove that? How have you been able to show that these things are happening?
So, okay, first I got to say, probiotics are for healthy people. You're not allowed to say they're for a disease. But with that said, I actually, so Parkinson's and Alzheimer's are in some publications known as type 3 diabetes. So it's an insulin resistance of the brain where the glucose isn't getting into the brain. But the idea for sugar shift actually originally did not come out of a concept of necessarily changing sugar metabolism. I went to a Parkinson's conference and there were some researchers there presenting a paper where they showed that the sugar alcohol, mannitol, could stop the aggregation of the proteins involved in Parkinson's. So I came back from that meeting. I got the paper. I read it. I was like, wow, this is super interesting. They were trying to get just people with Parkinson's to take oral mannitol. And of course, I had been looking at the microbiome and thinking about like loss of function. And so I went out and I bought this little book on mannitol. It's a little skinny book, chemistry, uses, and potential side effects of mannitol. But when I started reading it, I was like, wow, this is one of the most amazing molecules. But one of the first chapters in the book is utilization and production of D-mannitol by bacteria. And I was like, whoa, okay. So this was really where my concept of all our formulas came from, in terms of going, oh, well, bacteria make these things by converting something else into this. Can we put the factory workers back in the gut? And so I had a friend who was a fermentation chemist, Steve Cosme, and we got together and I was like, I want to make a formula. Can you like make me a prototype to try for John? And, you know, we had been measuring John's. I mean, he has hundreds of microbiome samples tested. And so we made this small batch of like 100 bottles, and John started taking it. And we could see this huge difference in his walking, how he was feeling, his energy. And then we sent the samples off to test, and we did testing of samples for 120 days. And we could see that his system was continuing to remodel for the full 120 days. And a number of the pathogenic bacteria in his gut that were also in some of the early Parkinson's papers were just like disappearing, like streptococcus. And so I was like, okay, and the way that the bacteria in the gut make mannitol is they're converting glucose and fructose into mannitol. And it's also a self-balancing pathway. So like if you make too much mannitol, it'll reverse and make the other. So, you know, you're not going to get too much of a good thing, I guess I would say. So, you know, we just started having people test it and try it. And then we did some small studies. So we did a study with just 6 people, looking at them for a month. And we saw that it was shifting the microbiome. They were making more butyrate, which feeds the gut lining. So we knew that was good. We had a 60% drop in antibiotic resistance genes. And we had an increase in siderophores. So it was doing something related to iron, which I knew was also a problem in Parkinson's. So, you know, we did that study. Then we did another little study with like 3 people who had IBS, IBD, and there were some published species of bacteria that were biomarkers for that. And we saw in 60 days that those biomarkers were decreased. And so we're like, wow, you know, this is really interesting. So we just kind of started like giving it out to people to try. And, you know, that's when I had some people with blood sugar issues who were like, you know, this has changed my blood sugar. It's improved my fasting blood glucose by, you know, 5 or 10 points in a pretty short period of time. And, you know, like somebody I went to high school with took it and wrote me back, and he's like, you know, my A1C dropped from, I can't remember the exact, but it dropped like 2 points over the course of like 6 months. And so, you know, we had been talking about whether or not we would try to do a clinical trial in Parkinson's, which I would still like to do. But, you know, Parkinson's it's a pretty complex endpoint measurement. You can't find, there aren't any really great endpoint measurements for really trying to say whether something has efficacy. And so we decided to do a clinical trial in diabetes because it has very clear, you know, fasting blood glucose, postprandial blood glucose, insulin, HOMA-IR. We did triglycerides, and we also did a safety study as part of that. So we did some kidney measurements. And then I wanted to look at a measurement called serum LPS, which is lipopolysaccharide or endotoxin. And that is coming from the gram-negative bacteria in the gut, which we knew there's a too much gram-negative action going on in the gut. And when those bacteria die, they release their endotoxin. Of course, every time you eat and a colony grows, it grows, goes through an exponential growth phase, and then it dies off and you're releasing endotoxin. And if you look in the literature, there's over 500 studies using endotoxin to cause Parkinson's, using endotoxin to cause depression, using endotoxin to cause diabetes. I mean, there's probably more. Those are the three that I've looked at. And so we measured serum endotoxin, and that was one of the most dramatic changes that we saw in the 90-day study. Then we unblinded the double blind at the end of the study, and we took a smaller group and kept them on the product for another 90 days. So, and over the course of 180 days, we saw a 14% drop in HbA1c. But the mechanisms of action are, we put a team together that works together in a sustained way to produce these metabolites.
So were those results based off taking once a day or twice a day?
Twice a day. So one in the morning and one in the evening.
Okay. And so go into the team of bacteria and the different roles that some of them, we don't have to go through all the, I believe there's 8, everyone does, but kind of highlight some of the big players in that.
So in the sugar shift formula, I like to think of it kind of as a fermented food in a capsule. And people actually do make yogurt out of it. But so it has 4 strains that either we or our partner Steve had isolated and done the work ourselves. So one is a leuconostoc mesenteroides. That's a mannitol producing organism. And that is typically found in fermented food, many fermented foods around the world. And it's sort of the first microbe in a fermented food that starts to grow and produce metabolites that feed the other good organisms in the fermentation that will produce things called bacteriocins that keep the bad bacteria out. So, when you have a good fermentation, you don't get any mold or, bad bugs in the fermented food. And it's because of the metabolites that these bacteria are producing. So leuconostoc is a mannitol producer. It's a butyrate producer. It's also kind of has some metabolites that can affect immunity. Another one of our organisms that we isolated ourselves is a Pediococcus acetolactici. Now, a lot of times you'll find Pediococcus in like fermented meats. So somebody like Paleo Valley with their fermented meat sticks, it probably has a Pediococcus in it. And Pediococcus is one of those strains that's a bacteriocin producer that targets those pathogens. And we found a Lactobacillus plantarum actually from fermented elderberries that came from my neighborhood in Colorado. And that Lactobacillus plantarum was resistant to glyphosate. Most Lactobacillus organisms are killed by glyphosate. And so with that plantarum, we started to look at it and study it, and ultimately found that it has something called the third pathway for glyphosate remediation. So it breaks glyphosate all the way down. And I think if I remember correctly, one of the byproducts of that breakdown is actually glycine, which we know glyphosate is a glycine analog. So we're able to break that all the way down and get back to the glycine. Wow. We have, and then the 4th strain that came from my partner Steve actually is a Lactobacillus reuteri. And reuteri is the the species that Dr. William Davis of wheat belly fame and supergut talks about all the time, because that's a organism that we've lost from our guts, but probably maybe 50 or 60 years ago, most people had Lactobacillus reuteri, but now we don't. And that's an organism that also produces bacteriocins, but it colonizes the upper GI tract and keeps those something called SIBO at bay, that's small intestinal bacterial overgrowth. So that the Lactobacillus reuteri is very important for kind of shaping the terrain of the small intestine and keeping the bad guys out, which you need because the small intestine is where we absorb a lot of our nutrients. And Lactobacillus reuteri has also been shown to increase the production of oxytocin in our body, and that's sort of the love hormone that sits at the top of the hormone hierarchy. So, the reuteri organism is also one of the mannitol producers, as is the plantarum. And we have two different species of bifidobacteria, Bifidobifidum and Bifidolongum. And bifidobacteria they're also highly susceptible to glyphosate. And so, what we saw during COVID and also with people with Parkinson's is they're typically lower. People who don't do as well with COVID and people with Parkinson's typically have lower bifidobacteria. And bifidobacteria can produce something called plasmalogens, which in the last, I would say, 6 to 12 months, there's more research coming out about how important plasmalogens are for brain health. And then we have a Lactobacillus paracasei, which has some research behind it around helping to reduce inflammation and support better immunity. And then our last organism is it's a trademarked strain from Deerland called Bacillus subtilis, and that Bacillus subtilis has a lot of research behind it in terms of helping to build muscle mass. And while the DE111 strain, the one that we have in our formula, hasn't been studied in Parkinson's, there is a Bacillus subtilis that has been studied in Parkinson's and shown in an animal model to reduce tremors. But, we've put this whole system together to kind of cross-feed each other, if you will, and make this sustained production that lasts for about 12 hours. So based on our computational modeling, we can see that it lasts, kind of making that mannitol and cross-feeding each other, what they need to be happy for about 12 hours. I like to kind of equate the formula to, we're trying to put part of the factory back in the gut, but, we want, if you're making a car, you want people at different parts of the line with different skills that can, put the pieces together. And then you want kind of your supervisor who's kind of overseeing it all. So I like to sort of think about it like that.
Martha, how do we know, or how can we be sure that the bacteria is making it, say, through the stomach and into the intestines to be able to do these roles? How do you view that? How do you, answer those questions? And I guess, I can only imagine what it's like to be in the supplement world and the probiotic world, where the marketing and the messages and all that goes back and forth. So how do you line that out?
It's a crowded space, I'll say. It's a crowded space of a lot of products that are very similar. Not similar to our product, but very similar to each other. Well, so first of all, you do get people who sort of sell you on, we have a specialized capsule that gets it all the way to your colon. Well, okay, what did I just tell you about reuteri? you need reuteri working in the small intestine.
Small intestine.
So if you have something that's not going to release until it gets into the colon, you're not helping the small intestine. And that's kind of interesting to me also, because there's 2 fecal transplant studies in Parkinson's, one that came up from the bottom and the other that went down from the top. And the one that went down from the top was successful in reversing symptoms. The one that came from the bottom was not successful. And my personal feeling about that, I don't think that they really address that in either one of these papers, is that probably with Parkinson's, there's some level of SIBO where those bad bacteria are in the small intestine. They're stealing the nutrients. I know they're making methane and hydrogen sulfide, and that's a problem. So we want our formula to be working all the way through the GI tract. And the other thing is, we're converting glucose and fructose, which we have far too much of in our diet. And those fructose and glucose gets absorbed in the small intestine. So you want to deal with that problem up here, not down here. I mean, you're going to get action all the way through, because as they produce their metabolites, in fact, what we showed is that it actually feeds a broader population of bacteria in the gut, and you get a healthier overall microbiome. We're working on that paper right now, but we had some of the keystone species that came back and flourished, like Roseburia inulavorans and Faecalibacterium prausnitzii, kind of a mouthful. But those are 2 keystone species that are often missing and also associated with blood sugar and metabolic issues.
Yeah. So on that topic, I know that Akkermansia has been studied as having a potential effect on blood sugar. So that, in my understanding, is an anaerobic bacteria that is, basically almost impossible to supplement. So what is going on in that context? Is that something that you pay attention to, that you think it matters? Is it something that having some of these more keystone species come in and cultivate the gut, that it happens to increase by, just the natural cycle of things? What are your thoughts on the other ones that we're not supplementing with?
So, Akkermansia is an interesting organism. And early in the microbiome research, there was a lot of people focused on that. People who were, I think, more thin maybe had more of it. There was a lot of, and one of the, I'd say the most published and well-respected researchers, Patrice Connie, who I think did work with Jeff Gordon at WashU and then went back to Europe. He has a product with I think it's heat killed. It's basically not live Akkermansia. I don't think it's available here in the United States, but because the other thing people don't, when we talk about probiotics and, what the CFU count is or how many live organisms you can get out of it. Well, microorganisms have action all throughout their life cycle. So while they're living and producing metabolites, they have certain actions. When they die, their cell wall components, they're peptides, and they have signaling actions. So, you see now people coming to market and saying we're making a postbiotic. Well, that can be, there's still not a great definition of it, but, what I'll tell you is when somebody grows my strain, it's a probiotic, a prebiotic, and a postbiotic, because it has live cells, it has, some cell wall components that are postbiotic, and it has, whatever metabolites were in the media when, at the point at which it was pulled off and dried. And So, there's just a lot more to bacteria than just the live bacteria. But, let's back up to Akkermansia again. It's a really interesting organism. And when I first started reading about it back in 2015, 2016, I'm like, ooh, we need to get one of these. And we talked about it a lot, but didn't do it. But as it turns out, Akkermansia is, it's elevated in both Parkinson's and multiple sclerosis. And it is what's called a mucin degrader. So it eats mucin. So if you have some issue in your GI tract where it's not getting enough to eat, it can eat the lining of your gut to feed itself. So if you start to really dig into the research around that organism, it's kind of one of those razor's edge things. You want like maybe just the right amount of it, but I don't recommend to people supplementing it because I've seen enough of the research on the kind of negative side of it that I think you have to be careful. Whereas, organisms that have been in fermented foods and we know they're not harmful, have been around for a very, very long time. So, but there are some people bringing some newer species to market that we've lost. And, I think there's some interesting things around that. But I also have a bit of concern about the way things are coming to market now, because the big capital that comes into the market wants something that they can own. And so they genetically engineer microbes, because you can own something that's been genetically engineered. You can patent it and own it. And a naturally occurring organism, you can't. Now, we were fortunate that we actually patented the formula that's, the mixture of the use of microbes to produce mannitol and as a potential treatment for diabetes or Parkinson. We list a lot of those things, and we were able to get a patent for that. But it's fairly unusual to get a patent for natural things that naturally occur in nature. Now, this particular 8-strain formula doesn't occur to get, or there's been it's not been shown that all of those are naturally occurring together in, any place that someone has looked. So that's partly why it was considered novel. And what we showed was that the pediococcus that we had added to the formula actually increased the production of mannitol and the conversion of those sugars, where, you could look at each of the individual strains and what they produce in terms of mannitol. And then, the combined group produced more than if you just added up the individual strains. So it's that synergism of the microbiome and the right team. I mean, it's just like if you're running a company and you hire the right people, you get incredible synergy that you can just do just about anything. And that's kind of the way I think about the microbiome, is if you have the right team and the right terrain and you give them the tools they need, they can do just about anything. And if you start to look at what microbes can do, what they can break down, and what they can produce, they are amazing. Like most of the drugs on the market first came from microbes, and then they started synthesizing them chemically. But I have a book about this thick that is the Pfizer book of microbial metabolites.
Wow. The metabolite aspect it's so broad, it's hard to even wrap your mind around. But for our mutual friend Morley Robbins, he recommends sugar shift to like everybody, right? As far as anybody that's in the root cause protocol, like he's just in love with it. So what are some of those things that we're seeing over the years of this being around, the impact it's having on people's lives?
Well, I mean, that reduction in endotoxin is a huge thing because it impacts
Yeah.
so much. I mean, because that's an LPS is highly inflammatory. So any of the kind of inflammation related things are being helped by that. But some of the ways that I've looked at it too, because Morley's a copper guy, and over the years, he's been teaching me minerals and the importance of minerals, and I've been teaching him microbes and the importance of microbes. And so I'll go back and look, and I looked at our formula to see, okay, do our microbes have multi-copper oxidases? Because we know in the human body, right, we're missing the copper, and we're like, so do we have working multi-copper oxidase? Well, the formula has multi-copper oxidases, and it has bacterioferritin, so it's helping to recycle that iron. And so I think some of those mechanisms in terms of, the RCP and how people are looking at that. And so I did have, I have one, actually the wife is a nutritionist and the husband has Parkinson's. And she started making the sugar shift yogurt for him. And she had been monitoring his iron that was way too high for many years. And after a year on the sugar shift formula, for the first time, his iron was in a normal range, in a normal range.
Wow.
So the yogurt, when you make the yogurt, it's at a higher count. You're going to have higher live organisms. You're going to have more of the metabolites, kind of more of everything, depending on, how long you let it go. But
Will you go through the steps of making the yogurt? Can you do that? And that obviously the higher count is part of why that would be necessary, or something that a lot of people are doing. So, is making the yogurt easy? Is it really hard? Is it something that we should just be overwhelmed and scared of? Like, what are your thoughts on that? Just that process.
I mean, to me, it's really easy. I actually first started making it without even a yogurt machine. So and if you think about, 100 years ago, people didn't have a yogurt machine. They were fermenting it, whatever the room temperature was, or if they had maybe sit it on a warm, something that by the hearth that's a little bit warm or something. But so, I mean, I first started out. So Dr. Davis is pretty focused on high fat in the yogurt because you need those fat soluble vitamins. And so I make it typically with half and half. Sometimes I even make it with cream. And that is, it's like almost too yummy. But yeah, because then you can eat too much of it and get fat. But anyway, I make it with half and half. But people do make it with non-dairy milks. It's just a little more complicated. But to make it with a whole milk or a half and half, I mean, I just open up the capsules. I mean, I'm not even preheating the milk. Now a lot of people will preheat the milk and they heat it to about 165 and leave it there to kind of heat shock it for a few minutes and then drop it down and chill it. But you can't put the microbes in it until it's cooled off to like 100 degrees. So like for me, I don't even do that process. I just open up my microbes, or we now have it in a powder form. You can get the yogurt starter, but I'll just open up. I mean, I usually make mine with 2 to 4 capsules. I sprinkle them over. We already have inulin in the capsule, so you don't have to add inulin. Some people will add more inulin, but I just put them in there. You want to make sure that your whisk that you're stirring it up with is clean, sterilized, because you don't want to contaminate it with yeast or any other bacteria from your kitchen. And then put a top on it. And when I started out with just a glass bowl and putting some plastic wrap over it and letting it go for 36 hours, and it's great. But I actually do use the Louvel yogurt maker now, and I typically
Yeah.
set mine anywhere from 98 to, I mean, you can go all the way up to 105, 106, but Lactobacillus reuteri is heat sensitive. So above 106, the reuteri will die. So I
Okay.
typically do mine closer to body temperature. So I'll do, 98 degrees, and I don't always let it go for 36 hours. I kind of watch it because you can get more whey separation the longer it goes. So, but it's not hard. I don't think it's hard. I do get a lot of questions from people because people want it to be like really precise. And it's more of an art than a science. And
Yeah.
what you find as you start to make the yogurts is at different times of year, you may get a different consistency. So, you might get more of the separation and whey, when it's warmer outside, or, when the barometric pressure is different. It's very
Yeah.
interesting, like people kind of reporting when they have, multiple bad, as they call them, bad batches in a row. But like sometimes I'll get a batch and it's more like cottage cheese, but it still tastes great. So I just strain off the whey and just make it into a soft cheese.
Well, that's cool. That's super cool. Yeah, we use the raw milk, and I've got the Louvel maker too. I've not made a batch with the sugar shift yet, but
So
I think that it's just fascinating.
I've
It's
done,
fun,
yeah, I've done the raw milk. To raw milk, you definitely have to heat the raw milk, and part of the issue there is the heating denatures some of the proteins, which is part of what
Yeah.
helps the milk coagulate. But I I have done it without heating it, but I feel like it's been a while. I can't remember it. Of course, you get the cream top that sits up at the top and doesn't really ferment the same way, and then you get a different consistency down below.
Yeah. I I love it. We do at the market. We've got the dairy that we work with. Joy, she comes out and she demonstrates making yogurt and butter and ricotta and does all this different stuff. And so it's really cool. It's just trying to bring this, uh, the science behind the health side and the homesteading type world together. I think is just a fascinating approach. And I homeschool my kids, so this is all. It's always time for some sort of a learning experience, and it's been fun playing around with all of that.
Well, on
Martha,
the
as we were,
one more thing on the yogurt, if you look, like Weston Price has a blog on GCMAF yogurt, and I actually
mhm,
put a recipe of it. So GCMAF, if you have not heard of it, it's GC macrophage activating factor. It's also known as vitamin D binding protein. And here in the United States, you used to be able to get, I think, a supplement of GCMAF, and doctors were using it for cancer treatment. And you can't get it here anymore. You can get it in Europe, but that yogurt, those yogurts make that vitamin D binding protein. So anyway, another kind of interesting tidbit there, which I think about, like vitamin A and retinol and Morley stuff, how important that is. And
yeah,
I think about that in the milk, but then I also take a step back on, the milk in general, and whether or not the nutrients in the milk, because the cows are eating the glyphosate laden grains and silage and stuff like that to produce the milk. So if you don't know exactly what the cow you're getting the milk from is eating, which is, hard to know. But I think with a lot of our commercial milk, part of that is probably that the glyphosate in the feed is impacting the quality of the milk that we're getting.
oh, absolutely, and it's affecting the cow's digestion for
Mm-hmm.
for sure. But I think that's all the more reason to work with and source from a local dairy that you actually know the person that is raising your food. I think that's
Right.
that's part of, the bigger message that we're, trying to get out there. So Martha, give me just kind of that basic do's and don'ts of who should use sugar shift, maybe who shouldn't use sugar shift, and the frequencies, with food, without food, once a day, twice a day, kids, adults, if you active cancer, just anything that may stand out there.
Sure. So, I mean, we do typically recommend that people take it with food because it works on breaking down the food and helping you access those nutrients. But, we do have people who fast, who take it fasting. It is our number one seller in terms of metabolic support. And, 80% of the population has some kind of metabolic dysfunction. So I think, generally we recommend it to just about everybody. We do have some other products, and one of those is called the antibiotic antidote. And I will often recommend that to people to start with if they've had a long history of using antibiotics or they've recently taken an antibiotic, because that helps with the kind of restoration of the ecosystem. And, take that for a few months. I mean, you can take that long term too, but then, probably switch to sugar shift to support your metabolic health. But all of our formulas have that lactobacillus plantarum that breaks down glyphosate. And I like to tell people, when they ask me, like, do you have to take a probiotic every day? Do you have to take this forever? And I said, well, typically you shouldn't have to take one all the time, but because we're exposed to so much glyphosate, and we know, we have a formula that helps reduce that, you probably do need to take something that's helping you with glyphosate on a daily basis. So, in terms of children, I mean, we haven't studied the product in children, but we have a lot of customers who do use it with their children. They're either doing the yogurt or, just opening a capsule and sprinkling a little bit on food. The research, there's quite a bit of research on probiotics in children and the benefits for that. Now, you want to be careful anytime you have somebody who's immunocompromised, even with probiotics, you've got to be careful with that. I do tell people if they are in cancer treatment, they really need to consult their pharmacist, somebody who really understands pharmacokinetics and all of that, because more and more research is showing how drugs affect the microbiome, but microbiomes affects the drugs. And so depending on, what you're taking, the microbes in your gut could actually break down that medicine and make it less effective, or they could accelerate it and make it, a higher dose than what you're really supposed. And a lot of people don't realize that. So anytime somebody asks me about, well, I'm taking this, and this, can I take your formula? And I say, you really need to go to your pharmacist and say, okay, this formula breaks down glucose and fructose and turns it into mannitol. And, this is the pathway. And, would there be any problems with the drugs that I'm currently taking? No,
I think that's a great point.
Yeah. And with my husband's, Parkinson's medications, there were never any problems with him on his Parkinson's medications. Now, he's now off of all his medications, and we're trying to kind of work through that. But there's a lot of side effects to those medications.
I think there's a lot of, unfortunately, side effects to all of them. But Martha, I just can't thank you enough for again, like the relentless research. Like it's greatly appreciated in bringing a solution to people to market. I think that's massive, to take the adversity that we're given and turn it into something to just try to help people, right? I think it's so admirable, and appreciate your friendship, and especially taking the time to share your story and the overall mission.
Well, thank you for having me. And I really love what you're doing, because the importance of real, whole, local food cannot be overemphasized. So what you're doing is really important work too.
Thank you for joining us in the Sowing Prosperity mission. If you like this content, like and subscribe, share with your friends. Be sure to check out sowingprosperity.com. Sign up for the email. We've got so much more work to do.